Welcome everyone. This module focuses on study design and quantitative thinking. We will have 5 sessions, the first 3 on study design and the last 2 on statistical inference. So, let us begin with the first session of the study design part of this module. So, it is useful at this point to quickly go over the scientific process involved in asking and answering questions in ecology. So, here are the broad steps. Based on our motivation, we frame a question. So, we may either be motivated by wanting to understand fundamental questions or wanting to address applied problems. Whatever our motivation, we have to frame questions very clearly because the effectiveness of study design depends on that. For certain types of questions, we may also need to frame one or more hypotheses and predictions from these hypotheses that allow us to test these hypotheses. Now, based on our question or on these predictions, we then design our study, go out, collect data, analyze and interpret these data, and then see where we stand relative to the question. So, how well have we answered the question? Maybe we have ruled out some hypotheses, but there are others we have to go into more deeply and so on. Here it is important to note that carrying out a study is an iterative process. So, it requires many iterations before we can come up with solid satisfactory answers to our questions. We in our project may only carry out one iteration, but others will then continue to build on that information. So, what we are going to do today and in the next couple of sessions is focus on study design. Now, let us also quickly go through the broad types of questions, that we may be interested in ecology. So, we might want to ask a question like, what is the population density of this butterfly species in this forest? So, here what we are setting out to do is asking a question that is about description. We want to describe a pattern. Or we might be interested in asking, why is its density higher in some forest patches than in others? So, here we are interested in looking at the process underlying a pattern. So, here we are interested in coming up with explanations for a phenomenon. Finally, another type of question that we might be interested in is how might climate change affect forest butterflies. So, here we want to forecast. To address these types of broad questions, there are some common basic tasks that we might be interested in. So, one task might be to measure a quantity. So, for example, we might want to measure how many species are there in a particular habitat, say a grassland. So, for example, we might estimate that there are 23 species in this example here, or we might want to compare butterfly species richness in 2 habitats, maybe a habitat that has experienced fire, so a burnt habitat and an unburnt habitat, and we might want to then come up with estimates like, for example, here 23 species in burnt habitats and 32 species in unburnt habitats. Another kind of task that we might be interested in is to understand a relationship. So, to correlate 2 variables. So, how is the number of species related with fire frequency across different habitat patches? Now, both in this comparison question and this correlate task, very often we are not just interested in describing this relationship, but we are actually carrying out this task because we are interested in understanding the process underlying a pattern. So, we might be interested in a causal analysis. Does fire affect butterfly species richness. Now, for cause effect kinds of tasks, it's very important to pay attention to certain elements of study design so that we can strengthen such cause effect conclusions. So, during these sessions we will also focus on elements of study design that will help us strengthen a causal analysis. Okay, so we have looked at some of the basic tasks involved towards addressing broad types of ecological questions. The next part here is to design our study. So, let us first try and understand what are the goals of effective study design. That is, if we design our study well, what do we hope to achieve? One of the main goals of good study design is generalization. That is, we want to generalize from sample to population. Let us look at this with an example. So, imagine that my question is, are male and female deer of a particular species, say barking deer as shown in this image here, are male and females of a particular deer species similar in size, or do we see size dimorphism? Are males larger than females, for example? To address this question, I go into the population, I sample 30 males, I sample 30 females and measure their body sizes. Now, when I estimate body sizes of males and females and compare them to answer my question, I am not just interested in those 30 males and those 30 females in my sample. What I want to do is generalize from sample to population. So, for this it is important to know what your population is that you are trying to generalize to. Next, reliability is another important goal of effective study design. So, what does reliability mean? How confident are we in the result obtained through a particular study design? That is, if I were to go to this population again, what kind of a result do I get? Suppose in the first year I carried out my study and I found out that males are 20% larger than females. Now, if I go back the next year and carry out my study, and if ecological conditions are roughly the same, if my study was designed well, I should get a very similar answer. If I get something very different, now if I estimate that females are 30% larger than males, then there is a problem in my design. This is assuming that ecological conditions are roughly the same. Okay. A third goal of study design is causal inference. So, typically when we study a phenomenon in ecology, we are not just interested in description, we are also often interested in trying to understand the process underlying a pattern. Now, for strong causal inference, ideally we need to carry out experiments. So, there are elements of designing good experiments. However, very often in ecology, we may only be able to carry out observational studies where we cannot experimentally manipulate conditions. So, how can we strengthen causal inference in observational studies? We will go through that. Finally, a last goal of effective study design is to ensure that we are actually answering the question that we framed in the first place and not something that is kind of related or adjacent to the question of interest. Okay, so now with these goals in mind, let us look at some key elements of study design that can help achieve these goals. The first element that we will talk about now is replication. So, if I were to tell you that I based my conclusion about whether males and females of this deer species are similar or very different in size, I based it on measuring one male deer and one female deer, you would not be happy with that study design, right? And that's because typically we want to measure multiple replicates, and the reason for that is because typically there is a lot of variation in ecological systems. So, we replicate to quantify this variability, which will then allow us to increase reliability and allow for better generalization from sample to population. The important thing to think about here is what is a replicate. A replicate should be independent. What we mean by that is that replicates provide independent pieces of information. If I were to measure one replicate, the next replicate that I measure shouldn't be correlated with that first replicate. That is what we mean. A second point to note here is that the entity or the unit on which we make measurements might not actually be an independent replicate. Let us try and understand this through an example. What is an independent replicate? It depends on the question. Let us take up this question. So, what this image here shows you is male blackbuck antelope gathered on a mating ground on which males defend tiny territories that are clustered together. Males then display on these tiny mating territories. Females visit these clusters, mate with the male and leave. The question is, why do males cluster their territories? It is very expensive, costly for males to do so. There is a lot of aggression between neighboring males. Males do not have any food on their territories, they have to leave their territories to feed. They lose a lot of body condition through energetic displays and so on. So, the question is, why cluster? Why not carry out the more common mating tactic in antelope, which is to have dispersed territories far away from each other with much less aggression. So, these dispersed, solitary or loosely clustered territories. Now, let us say I want to test the hypothesis. I want to test the hypothesis that males cluster their territories because females prefer to mate with males in clusters. So, this female preference selects for clustering in male territory. So, that is my hypothesis, and one prediction from this hypothesis is that male mating success increases with cluster size, that is with the number of males in a cluster. To test this prediction, I select 3 clusters, a cluster of 50 males, a cluster of 15 and a cluster of 5. Now, in the cluster of 50, I sample 10 males, I observe 10 males and record how many females visit and mate with each of these males. So, I get male mating success estimate for 10 males in the first cluster, 5 males in the second and 3 males in the third. So, my question now is, how many independent replicates do I have? I have measured 18 males, but do I have 18 independent replicates? Well, no, I do not, and the reason for that is because when we look at our question, we are testing the prediction that male mating success, here shown on the y-axis as the mean number of mates, varies across clusters of different sizes, that is clusters with different numbers of males. So, although I am making my measurement on males, I am following individual males or individual male territories and looking at mating success. Multiple males from the same cluster are subsamples that allow me to get a good estimate of mating success of a male for that sample. So, the scale of the question is across clusters. I want to know how something is changing across clusters, here male mating success, and an independent replicate here is the cluster. Take a different question. Suppose I am interested in whether central territories are more attractive to females, that is, do they use central territories as a cue in mate choice. So, my hypothesis is females prefer to mate with males on central territories, and my prediction here is that male mating success decreases from the center of the cluster to the periphery. For this question, I now measure male mating success, number of mates for a male on territories at different distances from the center to the periphery of the cluster. So, for this question, the scale is within a cluster at different distances, and here an independent replicate is a male on a territory. So, although my measurements for both questions were made on individual males or territories, in one case the independent replicate is the cluster, and here the independent replicate is the territory. Of course, I have to mention here that here too you can have non-independence in terms of adjacent territories affecting each other, and we will explore this point further now. So, this is the same figure as before, looking at how male mating success varies across cluster size, across clusters with different numbers of males, and what I have done here is color coded clusters based on their location. So, this is a cartoon map that shows the locations of these clusters, and you can see that some clusters are closer to each other than others. Now, notice that these two clusters here both have greater than average male mating success, and they are both also close to each other. Similarly, these two clusters here, orange, are again lesser than average male mating success, and they also happen to be in the same area. Now, it is possible that these two brown clusters have greater than average mating success, perhaps because they are close to a route that females use while moving through the landscape. So, now these two clusters are not truly independent replicates, they are both affected because of some environmental feature that they share. So, such correlations in space due to shared environment features or due to clusters affecting each other, in this case data points affecting each other, such correlations in space are very common in ecology. Another kind of correlation that is very common is time. Imagine that I made all the measurements on small clusters in the first part of the mating season between 2nd and 30th of March, and all my measurements on large clusters in the second part of the mating season between 1st and 30th of April. If I find a difference in male mating success between small and large clusters, I would not know, is it because of cluster size or is it because of time, because there are differences in females coming to mate in the first and second part of the mating season. What I need to do in this case is adopt a very important second element of study design, which is interspersion. That is, I should intersperse my replicates for small and large clusters through time. I should sample some territories on some small and some large clusters in the first part of the mating season, and I should carry out sampling in both small and large clusters in the second part of the mating season also, and in that way break up this correlation with time. Apart from space and time, there are other reasons why replicates may not be independent. For example, imagine that I am interested in looking at how food abundance in the environment around nests, how does that affect parental provisioning of nests, that is how much food they bring to nests, and chick growth. So, to do this, what I do is I monitor several nests and measure the mass of these chicks in areas with different levels of food abundance. So, what I have shown you here are measurements of 10 chicks from 3 different nests. So, chicks from a nest here are color coded the same. Because chicks share the same nest environment, the same parental behavior, similar genetic environment also, chicks from a nest may not represent independent data points. The good thing is, if we ahead of time, if we are aware of these potential sources of non-independence, these correlations in our data points, we can account for them in our study design and statistically. So, the reason we need to account for them in our study design is because we need a good sample size to capture variability, right. And if we have such correlations in data points, we have to measure a larger number of chicks in this case, or larger number of clusters, or larger number of males within a cluster, so that we can account for 2 data points not really providing independent pieces of information. If we measure these potential sources of non-independence, like for example, for spatial correlations, if we get GPS locations, if we get spatial coordinates, we record dates and times, we record nest identities, these can actually be incorporated for in statistical methods. The important thing is to be aware of them and incorporate them into design. Okay, let's consider a last example for this point about independence of replicates. Let's imagine that I am interested in the question of whether logging, that is the felling of trees, whether logging in a forest affects recruitment in a bird species, that is affects chicks successfully fledging to join, say, the juvenile or adult population. So, I am interested in how felling of trees, logging, affects recruitment in a bird species. So, what do I do for this? I select a logged forest, which I have just labeled here as F1, and I select an unlogged forest. Now, in the logged forest, I select 60 nests and monitor them carefully and record the number of chicks that successfully fledge from that nest. I do the same thing in the unlogged forest. Here are the data from these measurements. So, I get a mean of 0.8 chicks and a 95% confidence interval of 0.2 to 1.3 chicks per nest in the logged forest. In the unlogged forest, I get a mean of 2.2 and a 95% confidence interval of 1.7 to 3. Now, remember, confidence intervals are telling you what is the plausible range of values that true population mean. The recruitment in the unlogged, the recruitment in the logged are likely to be. Okay. So, now based on these data, my question to you is, can I conclude based on these data and this design, can I conclude that recruitment in logged forest is lower than recruitment in unlogged forest? Look at the data. The means and confidence intervals clearly indicate that these are different. But the question is, based on that, can I conclude that recruitment in logged is less than recruitment in unlogged? I cannot do so. I can definitely conclude that recruitment in F1 is less than recruitment in F2. The means and confidence intervals are clearly separated. However, I actually only have a sample size of 1 for logged forests and 1 for unlogged forests. So, I can make the statistical inference that F1 is less than F2. But there are many different reasons why two data points can be different from each other, right. So, I do not have replication at the scale of forests. So, statistically I cannot conclude that logged forest recruitment is less than unlogged forest recruitment. To make that inference that the difference between F1 and F2 is because of logging, I need to incorporate features of F1 and F2. I cannot do that based on the current study design and statistics. What I have is a case of pseudo-replication. Multiple nests in F1 are subsamples that go towards getting a very reliable, precise estimate for F1 and similarly for F2. Ideally for this question, I should match replicates and statistical inference to the question, which means forest patch would be an independent replicate and I would need multiple logged and unlogged forests. But for such large scale questions, very often replication is not possible. It may be very difficult to measure lots of different forest patches, Large scale questions are important. Many questions in ecology are at that larger scale. So, the question is, if we cannot replicate at that scale, what can we do to strengthen our conclusions? First, we could measure potentially confounding conditions. So, if we measure conditions that are important for this bird species in F1 and F2, we can show that these are roughly similar between F1 and F2 and the only real difference between F1 and F2 is logging, whether they are logged or not. Second, if for at least one of the categories we could have multiple replicates, that can strengthen our inference. What do I mean by this? Imagine that what I have shown you here is, imagine that these are the estimates I get from measuring one logged forest and one unlogged forest. Like I mentioned, I do not know whether this is a true difference or something that happens just by chance, because there are so many things that could be different between F1 and F2, including just stochasticity. However, imagine that I am actually able to measure multiple replicates for unlogged forests. Say I can measure 3 replicates. Now, if all 3 replicates in unlogged have higher recruitment than logged, this strengthens my conclusion that logging can depress recruitment. On the other hand, if I find a lot of variation in recruitment in unlogged forests, then I can conclude that in fact logging does not appear to affect recruitment in this bird species. A third thing that I can do when I carry out large scale questions without much replication is make very clear where statistical inference is and where biological inference is. I can also collect additional information and show that the sample is a good representative of the population. That is, F1 is a good representative of logged and F2 of unlogged forests. Next, I can really strengthen my conclusions if I test multiple lines of evidence, test multiple links in my hypothesis. So, for example, if the way in which logging affects recruitment is that logging reduces the population of insects that are an important source of food for chicks. This results in parents not bringing as much food to nests, which reduces chick growth and which results in greater chick mortality. Then I can actually measure insect populations in both forests, F1 and F2, measure how much food parents bring to the nest, measure chick growth regularly and measure chick mortality. And if I find that all these align in the direction of my hypothesis, that provides much stronger evidence for my hypothesis, and I can make stronger causal inference. Finally, what we can also do is carry out a meta-analysis. That is, we can put together studies from different forests that have addressed this question. So, here our study is one data point in this larger meta-analysis, and we ask, do these different studies find similar results for the effect of logging on recruitment? Okay. So, to sum up, what we have looked at in this first session on study design is we have talked about two key elements of study design: replication, and talked about the importance of identifying what is an independent replicate, being aware of possible correlations in our data points and the sources of these correlations, thinking about pseudo-replication, thinking about large scale questions and how to strengthen inference in large scale questions. We have also talked about the importance of interspersion and how this can help deal with confounding variables like time and space. So, that brings us to the end of the first session on study design. Thank you.